So some of the audience present here may be familiar with ClonePix technology already, but today I wish to discuss a new application detecting GPCR target proteins on the cell surface. So today's discussion will focus on a three-platform approach to detecting, picking, and validating functionally active GPCR proteins. Primarily, the focus will be on the ClonePix system as a means for screening mammalian cell colonies in situ using both bright field and fluorescence, and then picking individual colonies into 96-well plates. The Clone Select Imager provides a means of monitoring cell proliferation to assess both cell health as well as determination of appropriate confluence for further validation assays. Once cells reach appropriate confluence, cells are treated with an appropriate agonist to activate the GPCR signaling pathway. The Flipper Tetra system measures associated cytoplasmic calcium changes using a calcium-sensitive fluorescence dye. Thus, this validates that positive signal as observed on ClonePix is indeed the functionally active GPCR protein of interest. Therefore, the three systems work synergistically together to provide a rapid and robust method to detecting GPCR proteins of interest. Prior to discussing the work done with detection of functionally active GPCR targets, I will discuss the fundamental technology behind the ClonePix and how it can assist your workflow. So what are the bottlenecks in typical cell line development? Conventional limiting dilution is cumbersome and is something I wish to never have to do in the lab again. Looking at the boxes of microtiter plates this man is sitting on, to screen 10,000 clones would be near impossible. Even a typical limiting dilution experiment requires significant incubator space, ELISA reagents, and the resources of your lab staff. Being able to accomplish that in less than one workday would significantly accelerate the number of clones one can screen, increasing the probability of finding optimal candidates. ClonePix already has replaced limiting dilution in hundreds of labs globally. Well, let's take screening by FACS. While there are advantages to FACS, cells are fragile to the high pressure required by firing down a capillary tube. It would be beneficial to have higher cell viability after spending all that significant amount of time and resources that are required by the use of a FACS system. Or, when screening for functional GPCRs, one may be in a situation where they need to perform different assays and utilize different technologies for each pathway or target. Luminescence, fluorescence polarization, radiometric absorbance, resonance assays, scintillation proximity assays, all to measure a cyclic AMP accumulation or IP accumulation or beta-arrestin binding or calcium changes or receptor binding. It's a lot. So although those standard functional assays may still be required at some point, it would be nice to utilize one technology and one screen to first verify the presence of your target receptor. Which brings us to the ClonePix. With ClonePix technology, cells are plated in a semi-solid media where they form distinct colonies with minimal handling. Picking is very gentle and collects a colony of clonal cells, not a single cell, so outgrowth is consistently high. This permits early separation of clones in a mixed population before they are swamped from overgrowth from other cells. Efficient removal of unwanted clones is done at the earliest possible stage. Up to 10 source plates, or the plates containing the cells in semi-solid media, and 10 destination plates of liquid expansion media can be loaded at once. Utilizing the stacker cassette visualized on the left, plates are placed in trays with a magnetic backing. The magnetic backing secures the plates, and then the stacker is automatically raised and lowered in the system while one plate is removed for imaging and picking at a time. The system will image the plates and allow the user to modify morphological and fluorescent criteria to pick only the desired optimal colonies. The system will pick the colonies into 96-well plates. An automatic sanitization routine is completed in between picks to ensure sterility. As far as throughput, a 96-well plate can be picked in this fashion in less than half an hour, thus providing a throughput of imaging of up to 10,000 colonies an hour and picking up to 200 of the desired ones. Prior to committing to picking, one can utilize software tools to gate out undesirable morphological and fluorescent criteria. Colonies can be ranked by the desired statistic. By clicking on any one of the dots in the ranking plot, the associated colony appears as a thumbnail on the same screen, providing visual verification. A quick review shows exactly which colonies will be deposited where in the 96-well plate. Then the ClonePix proceeds to sterilize the pins and then independently fire and retract the pins as it gently collects the colony in the 6-well plate and then deposits into 96-well plates of expansion media. Now that the fundamental instrument process has been discussed, what is the basis of plating in semi-solid? How are these target proteins detected? For the next few minutes, I will discuss how this is achieved, and then I will discuss how GPCR target proteins are detected. Traditionally, the ClonePix technology was developed to detect secreting antibodies from mammalian cells. Individual cells are plated in a semi-solid media. This media allows the cells to proliferate and form colonies while being held stationary and distinct from neighboring cells. Because cells grow as individual colonies, many different cells can be plated in one well, thus increasing the stream throughput to many different cells at once. Fluorescent labeled antibodies are also plated with the cells. In this case, the cells are secreting immunoglobulin IgG. A 50-labeled anti-IgG, such as molecular devices CloneDetect, is small enough to diffuse through the semi-solid matrix. As the cells secrete IgG, the ClonePix binds to the antibodies, forming a complex around this colony. Using a different animation here, this captures the same principle of secreted protein aggregating around the colony. With capture probe present in the medium, the secreted protein and the capture probe are mutually trapped locally around the colony of cells, producing a halo effect. The amount of fluorescent capture probe is proportional to the amount of protein secreted. As visualized on the ClonePix, physical locations of the colonies are detected under white light, and then the secreted protein, or halo, under fluorescence. The software looks for fluorescence around the vicinity of the colony, visualized under bright field. While detecting secreted proteins has been the most common application on ClonePix, the ClonePix is not just limited to this. ClonePix also has the ability to capture intrinsic GFP expression. For instance, here an MCF7 line was transfected with GFP fusion protein and grown under adherent conditions. The ClonePix system detected the colony in white light and fluorescence and it accurately picked the highest intrinsic expressing colony. In addition to secreted protein and intrinsic expression, we have recently validated that ClonePix can be used to detect proteins on the cell surface. Although the sensitivity of detection is limited to the number of sites on a cell surface rather than accumulated secreted protein over time, the ClonePix has the ability to identify cell surface proteins when adding a fluorescently labeled protein against the target protein. P2Y1 receptor belongs in a family of G-protein-coupled receptors and functions as a receptor for extracellular ADP and ATP. While this was imaged using an FNAP blue label polyclonal against the receptor, cells expressing multiple surface proteins or markers can be isolated by multiplexing with up to 3 fluorescent probes simultaneously. Finally, in the case where a directly labeled primary antibody is unavailable, it is possible to use a dual antibody approach using an unlabeled primary plus a labeled secondary. The concentrations of each antibody may need some optimization, but the complex can form with minimal nonspecific binding. So for example, this is an example of a dual-labeled antibody screen under white light, and then also visualized using the rhodamine channel. Knowing that ClonePix has historically been successful with detecting secreted and intrinsic proteins and having some preliminary ideas it had the sensitivity to detect cell surface proteins, we wanted to determine if it was feasible to detect GPCR target proteins on the surface and verify that those are indeed functionally active. So GPCR proteins, what are they? This represents a generic model of deep protein-coupled receptor, or GPCR, signaling pathways. GPCRs are also known as the 7 transmembrane domain receptors because they pass through the cell membrane 7 times. They constitute a large family of receptors which sense molecules outside the cell. Once ligands bind and activate those receptors, this activates signal transduction pathways, resulting in cellular responses. These ligands include hormones, neurotransmitters, pheromones, and light-sensitive compounds. Essentially, an agonist activation of a particular GPCR activates a chimeric G protein, which in turn activates phospholipase C, which cleaves inositol triphosphate, or IP3. The IP3 diffuses to receptor sites on calcium channels in the membrane of smooth endoplasmic reticulum. When IP3 binds to the channel, it opens, and calcium ions flood into the cytosol, leading to cellular responses. So why are GPCR proteins important? GPCRs are by far the most extensively validated class of therapeutic targets, being up to 50% of all pharmaceutical drugs in the marketplace. And there remains tremendous potential for targeting new receptors and their downstream effectors. Of particular interest are the orphan receptors identified by the Human Genome Sequencing Project. Orphan receptors are receptors that do not have a known agonist or physiological function. Therefore, there remains a significant amount of opportunity to discover ligands to these orphans. Current methods employed in GPCR screening programs measure G protein signaling by determining changes in secondary messengers, such as cyclic AMP or inositol triphosphate or intracellular calcium mobilization. Recalling my earlier slide of all the different functional assays, this often demands setting up different assay platforms for each and requires special instrumentation or different kits for each pathway. Another challenge in typical screening assays is the assay requires orders of magnitude higher concentration levels of the target GPCR on the cell surface than endogenous levels. Thus, an agonist or an antagonist effect can greatly differ in certain functional assays compared to the endogenous level seen in the patient. Thus, a need exists to detect various expression levels across the different GPCR pathways in one system. This brings us to our ClonePix screening. For our feasibility study, we chose to work with the muscarinic receptor. The muscarinic cholinergic receptors belong to a family of G protein-coupled receptors. The M1 receptor is among a growing number of G protein-coupled receptors that are able to activate multiple signaling cascades. For the plating experiment, we chose a CHO-K1 cell line transfected to express the muscarinic 1 receptor, or the CHO-M1 line. Different passages were plated as we wanted to see if the ClonePix could differentiate amongst the different levels of expression. We plated the parental CHO-K1 as a negative control. We chose to also plate cells using 2 fluorescent detection methods. The first was with a direct labeled antibody. The second was using a secondary antibody against an unconjugated primary. The latter was to determine if, in the case a direct labeled antibody was unavailable against the target protein, a dual approach could be used with minimal background and non-specific binding. So our cells were initially picked on the ClonePix, and then they were monitored for proliferation by the CloneSelect imager, and then functional validation on the flipper system. So I'll take you through each of those 3 results. So on the ClonePix, we were able to differentiate between various expression levels. So keep in mind that traditionally, ClonePix has been used to look at an aggregation around a colony, which is a lot more binding than you would typically see on a cell surface. So we were wondering if we could actually see at that level of sensitivity a low or higher levels of binding to cell surface. And indeed, we were able to discriminate between various levels of expression. You can see that some of the colonies corresponding fluorescent images here are brighter than others. Morphologically ideal colonies were gated out from colonies that were determined as too small, irregular, or too close to other colonies. The remaining were ranked by interior fluorescence intensity and separated into high, medium, low, and no signal groups. A gallery view of thumbnails allowed us to quickly assess the groups the colonies had been placed in and verify that they were indeed what we wanted. This particular picture captures the thumbnails of all our high expressors. Results from our experiment demonstrate positive signal from both direct labeled and dual labeled approaches. And our parental cell lines failed to produce any significant signal in the rhodamine channel or the PE channel. With our dual-labeled approach, we did not observe any nonspecific binding, as observed from a lack of a signal in wells where only secondary antibody was added. This gave us confidence that a dual-labeled approach can be utilized as necessary. So the ClonePix results, our negative control or our parental CHOK1SC lines failed to produce any fluorescent signal. No fluorescence was observed in the wells that we plated without antibody, thus eliminating autofluorescence as a potential signal. The secondary antibody without the primary antibody failed to produce a signal specific to the colonies identified in white light. And also, the ClonePix was able to successfully distinguish amongst different expression levels of the M1 receptor. After picking the clones on the ClonePix, we moved to using the CloneSelect imager to monitor confluence. So the CloneSelect imager is a platform that allows one to image cells without any stains, labels, or dyes and monitor for confluence and automatically determine confluence, cell number estimation, and growth charts for every single well of your plate and track over time. So utilizing the CloneSelect imager, the clones that were picked from the ClonePix were deposited in 96 well tissue culture plates and imaged on the CloneSelect imager to verify transfer and also subsequent growth for 1 week after picking. The growth curve generated from the CloneSelect imager demonstrated proliferation of our picked cells. Also using the CloneSelect imager, we were able to determine when the cells were confluent enough for the calcium-6 assay on the Flipper Tetra system. And in fact, the CloneSelect imager is a beneficial tool anytime one needs to use prior to a cell-based assay to ensure that their cells have reached appropriate confluence levels. To validate that the fluorescence observed from ClonePix corresponded to a functionally active GPCR receptor, the Flipper Tetra system was used to evaluate intracellular calcium changes using the calcium-6 assay. The assay utilizes a calcium-sensitive dye absorbed into the cytoplasm. Once ligand receptor binding occurs, intracellular calcium is released, binding with the dye. This is quantified by increasing levels of fluorescence intensity readouts. For experimental methods, prior to the calcium-6 assay, cells were transferred from 96 well to a 384 well format and allowed to grow overnight at 5,000 cells per well. Then the appropriate compounds and dyes were added. To changes in cytoplasmic calcium via activation of the G protein-coupled IP3 sensitive pathway by carbachol was assessed at 40 nanomolar. Finally, fluorescence readings were taken both before and after the carbachol addition. In our high-tech clones, carbachol produced a 4-fold increase in fluorescence from our background. In our medium and our low-expressing clones, carbachol produced a 4-fold and a 2-fold increase in fluorescence. And finally, in our parental CHOK1 cells, there was a lack of response. In fact, note that the initial signal was only 20% above background compared to a 400% baseline in our actual expressing cells. So in our CHOM1 high fluorescent clones, carbachol produced a 4-fold increase in fluorescent rate from our background, and we also saw a 4-fold and 2-fold increase in our medium and low fluorescent PIC clones from the background. And then in our parental CHOK1 negative group, carbachol failed to elicit any significant change in fluorescent rate from background. Therefore, these results support a positive correlation between membrane-bound M1 GPCR expression level and the actual functional activity. The lack of calcium fluorescent signal in the CHOK1 negative control group confirms that the ClonePix system can accurately distinguish between clones with and without the expression of M1 GPCR on the cell surface. So in conclusion, we found that the ClonePix 2 is not just for looking at secreting proteins, but it can reliably detect cell surface proteins and reliably detect variable expression levels of those cell surface proteins. And it's an efficient means and one step for a high throughput selection of clones of interest. Utilizing both at white light and fluorescent images in situ, the ClonePix system has a sensitivity to detect endogenous levels of protein limited to the cell surface expression. And it can be used to efficiently detect and also pit respective GPCR expressing clones, which will provide a high-quality GPCR protein for a variety of applications. So with that, I thank you, and I want to turn the floor over for any questions that you may have. I received one question. For the functional response on Flipper, how difficult it is to transfer the clones. I suspect that means how difficult is it to transfer the clone from one plate to another for the read on the Flipper system. It's not difficult at all. It's just a simple pipetting step of just monitoring for confluence to make sure that they're viable and confluent enough, and then just transferring them onto a 384 well plate. Please type in a response if that does not answer your question. Any other questions at this time? I also have one other question. Did you identify some of the highest GPCR expressors with altered or lowered growth rates? We didn't do an extensive study to monitor proliferation amongst the highest lowest, but we didn't see anything that drastically was observing higher or lower growth rates amongst the expression levels, so no. Another question is, are all cell lines able to grow in a semi-solid media and form colonies? The ClonePix has been validated for many cell lines. I can't say every cell line has been tried out there, but we have done an extensive amount from HEC, a variety of CHOs, NBCKs, hybridomas, myelomas, a wide variety. All right, with that, I thank you very much for your time, and have a great day.